reading it busy. You studying, I am asking. Okay, online people, please confirm, am I audible? Okay, no problem, let's start. Okay, so we'll be discussing about CSIR NET June 2025 question paper. Okay, all right, continue. Okay, now every one of you remember my words very correctly. See, all of you remember my words, please. It's not necessary that you have to give the correct answer for every single question, but whatever the questions that you can do, you try to do it very accurately and try to analyze it very correctly. Okay, so always remember everyone, out of 50 questions in the part B, try to answer 20 to 25 questions very accurately, right? And when it comes to the part C, out of 70, I am using the word here only 20, okay, but 20 questions, make sure that you give a correct attempt. Okay, now out of 75 questions, at least I want you to give 18 to 20 questions correctly. Okay, so based on that we can understand the performance analysis. Okay, all right. Okay. Okay, everyone, let's continue. Good afternoon. Okay, so the question number 1. Okay, right or wrong, every one of you please try to give the answers. Okay, online people, everyone please be active and keep giving the answers. Even though you don't know it, give a try. I'll be explaining all the questions. What's the answer? 2 isomerase. Arams. Please come, please come. Okay, I think it's visible for online people. Please start giving the answer. The question says that which one of the following enzymes can unwind short stretches of the DNA helix immediately ahead of the replication fold? See, there is no doubt the answer is the helicase. I want you people to understand, whenever they are giving a word unwinding or unzipping, or when they use the word stand separation, every one of you try to remember it is definitely helicase only, right? But in the question, if they ask which releases stress, right, or which remove supercoiling, or which releases torsion, and that time we have to choose the topoisomerase. We know very well topoisomerase only called as the gyrase in case of a prokaryotic organism. In the CSIR official key, they have given the answer as DNA gyrase, which is absolutely wrong. The answer would be the helicase, because that is the one which is generally separating or breaking the hydrogen bonds and separate the 2 strands very easily, right? Okay, you will get the PPT and you will get all the explanations also, don't worry about it. Right, okay. Then which one of the following is an example of asymmetrical division in plants? First of all, tell me what is called symmetric division and what is called asymmetric. These are the terms which we read in the intermediate. Very good. What is that? See, very good. Symmetric division, nothing but what? Which give rise to the equal number of cells which exactly possess the characteristics of a mother cell or a parent cell. Whereas asymmetric division is a type of a division. Here also there is a production of the daughter cell, but they never work as mother cell, or even within these two daughter cell also they do not show the same characteristic features. What's the answer? So one is equal and one is unequal. See, this is just intermediate 11th standard. Remember all of you, first mitotic division of a microspore and megaspore. Both are generally symmetric division only. One mother microspore is going to give rise to what division? Mitosis, right? Which is going to give rise to the same cells, they also perform the similar function. Same thing will happen in the microspore also as well as in the megaspore also. And look at this, cell division in the root elongation zone. Even in the root elongation zone also, the cell divides and it gives rise to the many number of cells. They generally look like same and they also performs the same function. Even in the root elongation zone also, it is a symmetric division only. The only asymmetric division is what? Formation of stomata. Guard cells from a guard mother cell. If you might observe, see, these are the two guard cells. In between the guard cell we are having what? Stomata. Most of the time, I am using the word most of the time, these two guard cell structure will be different, right? And functions, they are almost different. In certain cases they are similar. But in a given 4 option, we can consider this will be the most appropriate option for the asymmetric division. So focus everyone quickly. Asymmetric cell division is a process where a cell divides into two daughter cells of unequal size and are different fates, right? In case of what we can say microspore, megaspore, root elongation zone, in all the 3 cases we are going to produce the cells which are completely identical. Only in case of a guard cells, these two guard cells are completely having a different shapes as well as different functions. So that would be the example of the asymmetric cell division. No confusion. This we read in the PPT itself, straightforward question. What is the answer? We read it now. This is called satellite cells. We already read it. Very recently also I explained you. We discussed, right, nervous tissue made up of 2 cells. What are those? Number 1 is called as neuron. Apart from neuron, what else we are having? Very good. Neuroglia. How many times I have explained you? How many neuroglia cells are there? Total 6. 4 are present in the CNS, 2 are present in the PNS. Correct na? What are present in the CNS? Remember? Very, very good. Astrocytes, oligodendrocytes, microglial cells, ependymal cells. Is it? And what are present in the PNS? Very good. Schwann cells, satellites. Correct na? So these 6 are generally said to be the glial cells. Among this, which is not glial cell? Which is the basket cell. It's a straightforward question. This is with represent or which is related to the cladogram. Good, good. Everyone, please. Artifact means a statement which is suggesting the relationship. It's very simple. They are asking a question. Actinobacteria and proteobacteria both are possessing a close relationship, or both are showing a close relationship. That we come into the conclusion that they are possessing the close relationship based on a 16 rRNA gene, right? Among these 4, which is majorly suggesting that these 2 are closely associated based on 16 rRNA. That's the question. Okay, no need to confuse. The answer is 1. Compositional heterogeneity causing long branch attraction. See, long branch attraction or LAB, which means that the two species are closely connected with each other, right? So, here they are closely connected with each other with respect to what? Complete heterogeneity. Here the compositional heterogeneity, the word generally represents they are having similar base sequences. Similar base sequences, especially in the actinobacteria and the proteobacteria, most commonly they contain equal amount of GC sequences. Right, now look into this. It is all theoretical only. Compositional heterogeneity causing long branch attractions. Here, long branch attraction is a phylogenetic artifact where rapidly evolving lineages are attracted to each other or to the outgroups, often resulting in incorrect topologies. In more simple words, compositional heterogeneity is a process here, differences in their base composition, especially in the GC, which leads to the long branch attraction. So that is only called as compositional heterogeneity. In some some species, GC sequence is similar. In some conditions, GC sequences differ completely. That is called compositional heterogeneity. That is generally explaining the differences or the close relationship between actinobacteria and proteobacteria. Whereas homoplasmy as well as inaccurate, first of all, inaccurate routing never ever explain the relationship between the two clades. If the routing is perfect, then we can understand whether these two species are related with each other or not. So homoplasmy, parametrization, these two methods are not explaining the relationship between these two. It was only majorly explained by compositional heterogeneity. Please remember whichever you don't know. Now you please count how many questions you will be able to solve. This is also directly there in the slides. It is directly there in the question. Which one of the following carotenoid is not involved in the non-photochemical quenching of the chlorophyll fluorescence in plant? First of all, what is called non-photochemical quenching? We read in the concept of photosynthesis in the first class. See, plants having certain photoprotective mechanisms, correct na? What is called photoprotective mechanisms? See, we animals, when we expose it towards the sunlight, we will try to escape because we can move. Plant cannot move it, correct na? So they have to tolerate to the excess intensity of light. So to tolerate the excess intensity of light, they are going to show certain mechanisms which are called as photoprotective mechanism. We all discussed it, which is called as first mechanism called photochemical quenching. Quenching means releasing the excess energy in the form of a heat. That is called quenching. Then second one, anyone remember? What is that? Correct. Now, what are non-photochemical quenching processes? We read in the plant physiology photosynthesis topic. Anyone tell me what are the other mechanisms? Another one is called scavenging molecules, such as what we can take, carotenoid, SOD. We all read in this slide itself, in the photosynthesis slide. So these are all said to be what? Non-photochemical quenching process. When it comes to the photochemical quenching process, releasing the excess amount of energy in the form of a heat. In this, all of the molecules are involved except one. One is zeaxanthin, vialoxanthin, anthroxanthin and canthaxanthin. So zeaxanthin is the major one among all. Vialoxanthin and anthraxanthin are intermediate molecules which are involved in the non-photochemical quenching. Canthaxanthin is not involved. It is so simple, straightforward question only. Non-photochemical quenching is a mechanism that protects the plant from excess light energy by dissipating it as heat. Dissipating means releasing it in the form of a heat. So what are all involved? Zeaxanthin is directly involved. Vialoxanthin and anthroxanthin act as an intermediate molecule. Canthaxanthin is not at all involved. Why? Because what I can say, vialoxanthin, zeaxanthin, anthroxanthin, these three belongs to the category of carotenoids, whereas canthaxanthin is not a carotenoid, so it is not at all involved in the photoprotective mechanism. So most of the students get confused with this question. Try to answer correctly. Think about it properly. Everyone, stereoideogenic acute regulatory protein facilitate the transfer of cholesterol in which of the following subcellular organelle? Now what's the answer? See, most of the students, since it is related to the cholesterol or a sterol, what we understand, we have endoplasmic reticulum, in that RER is involved in the protein synthesis, and we think that SER is associated with the lipid transport or the cholesterol transport. Okay, that is true, but the question is about a specific protein. The name of the protein is a star protein. The ultimate function of the star protein is what? Transfer of. So it is generally transferring the cholesterol into the mitochondria, right? So into the outer membrane of the mitochondria. Okay. Now remember, name of the name of the transporter protein, which is called as star. It is required for steroidal hormone synthesis. It facilitates the transfer of the cholesterol from outer mitochondrial membrane to the inner mitochondrial membrane, right? So in the inner mitochondrial membrane, cholesterol is getting converted into the presinol. So remember, major location of the star is on the outer membrane of mitochondria. Remember, even though you don't know, you remember. See, luckily in the 2025 paper, 6 marks came from geological time scale, which is very easy. Major eras from oldest to youngest. We already studied, no? Everyone quickly give the answer. Question number 7. Why you are thinking? I don't know. What is the answer? 4. Archaean is the oldest. Archaean, Proterozoic, as well as the Phanerozoic. Okay, you please try to, you are not reading, you try to remember. Archaean is the earliest era, spanning about what? 3.8 to 2.5. Proterozoic, 2.5 billion to 541 million. Whereas Phanerozoic is what? The recent era or the current era. It is just started about 541 million years ago. Write down, everyone, remember. They are asking oldest to the youngest. Archaean, Proterozoic, as well as the Phanerozoic. You always write down in that the same way only. Oldest to youngest you write down. Oldest, intermediate, and this will be the youngest according to this. Right. So this is completely information based. Only botany, core botany people can answer this if you might read it. Everyone try to give the answer for the question number 8. Just give it a try. It's okay. Tomato plants in a subtropical field begin to show progressive wilting, interveinal chlorosis, which is loss of chlorophyll, and root rot, damage in the root or lesions on the root. These are the symptoms shown by the tomato plant, which is living in the subtropical region. The subsequent laboratory analysis indicated that cenocytic hyphae, along with the presence of thick-walled oospores. See, what we seen is the symptoms. And when we do the laboratory analysis, we understand that all these symptoms are happened because of the presence of Sinusitic hyphae and the presence of oospore, definitely it is a fungal infection. Right. And what is the question? The question is, among all these species which is not involved in showing these kind of symptoms. Right. So the answer will be, you have to remember here, the answer is Phytophthora infestans. Very good. Okay. See why? Because Phytophthora infestans, it causes light blight disease on potato and tomato, not on the sugarcane. Right. So it shows lesions on leaves as well as the stems, but not on the root. Right. Remaining all these three majorly affect tomato plants. They cause loss of chlorophyll, which means remaining three species can easily act on the leaf also as well as on the root also. Okay. It is completely informative based only. Just straightforward. This is also from slides directly. Don't lose the confidence, you just try to analyze what you know properly. It's simple statement related. So simple question it is. Everyone online, what is the answer for question number 9? They're asking which is incorrect. Everyone, what is the answer? We read it no more. Forgot. What is the answer, Vijayant? Why? GAGs are distinguished by their sugars, the type of linkage between the sugars and the number and the location of sulfate group. First of all, GAGs are positively charged or negatively charged? GAGs are negatively charged. Correct? Na? Why they are negatively charged? Because they contain sulfate groups. They also contain the carboxyl groups. Because of that, they are negatively charged. Correct? Na? But they are linear polysaccharides, which are means what? They are completely unbranched. They are not the branched. They are one of the most anionic molecules made by the cells. Why they are anionic? Because of the presence of sulfur and carboxyl. They are unbranched. They are always followed by the repeating disaccharides such as, remember, sulfated amino sugar and uronic acid. They are always made up of repeating disaccharides. One is sulfated amino sugar, another one is what? Uronic acid. Okay. Severe loss of GAGs can lead to premature aging, which is true statement. It's only small statement which is unbranched here. Answer is 3. Correct, everyone is correct. Okay. This is also simple, straightforward question. See, population is generally divided into many demes or groups. So, population divided into subpopulation, population divided into metapopulation also. And the question is very straightforward. The question is about the Levin's model. Now, you always remember, Levin's model always explain about the stability. It always explain about what? Stability of the population. So, in most simple or specific word, you have to remember, stability generally represents rate of immigration should be equal to the rate of emigration. In other words, instead of using this immigration and emigration, we can use the word rate of extinction will be almost equal to the rate of recolonization. By that, the population becomes what? Completely stable. So, the movement in the given question, when you see the Levin's model, remember it is always talking about stability. Individual subpopulation should have realistic chances of extinction and recolonization. Okay, remember it is a fact. It is a fact based question. or which is already proposed statement. You have to remember it is always talking about the stability. This is also pretty difficult question. Try. This is all experimental. Look into this, simple. In a study on honeybee for aging, researchers investigated how dopamine release in the mushroom bodies affects the bee's ability to learn floral odors associated with a nectar reward. Which of Tinbergen's 4 questions does this study primarily address? See, in more simple words, to understand aging, the scientist named as Tinbergen, he generally proposed 4 principles. What are the principles that he has given? He explained mechanism, ontogeny, function, evolution. In more simple word, causation. Causation means how does it work or how does it happen? Development, how does it develop? Adaptation, how it became survive and what is their relationship, right? So what are those? Causation, which is called mechanism, development, function, evolution. These are the 4 principles of this particular scientist Tinbergen. So out of these 4 principles, any principle is present or not? So can you see the third one? What mechanism, which is called as causation. What mechanism underlie the association between the floral odor and reward in bees? So what you have to remember, what are the 4 possible explanations given by this particular Tinbergen. So remember, causation, development, adaptation and phylogeny. So in that one of the what we can say, one of the question is what mechanism underlies, means what mechanism is involved in the development. This is all a factual based questions. If you know the fact, we can answer. This is straightforward directly from the PPT only. We read multiple times this question. Many times we read it. The same lines only we read it in the PPT. Secondary metabolites. What is Jayanti? What is the answer? It is there in the first slide of the PPT only. Everyone, what is the answer, online people? See, the same line only I have explained to all of you. I don't know. The subclass of terpenes that are generally volatile and often stored in glandular hairs of epithelial cells, I have explained to you many times. Very good, everyone correct. Which is monoterpenes as well as sesquiterpene. Monoterpene and sesquiterpenes, most of them are volatile. We studied the same lines only, right? Clear everyone? Very good, all are correct. Same simple question it is. Sri Agna, what is the answer for this simple question? Chandini, what is the answer? Five times I explained. What is the answer? Everyone. Forgot. Why Jayanti? You forgot everything. We read PS1 as well as the PS2. Right? So between the PS1 and or PS2 to PS1, electron transport will occur. I have used certain herbicides and I have blocked this electron flow. Whenever I blocked this electron flow, what are the consequences they are saying? What is the outcome? PS2 is responsible for the production of ATP or NADPH? ATP. Correct, na? PS1 is responsible for the production of NADPH. ATP or NADPH is produced when the electron flow happened only. Now I have stopped this electron flow. Then what happened? Neither ATP nor NADPH would be produced. This is 11th standard question. The simple question. See, it's also straightforward question. Only every student remember what is called Cre-lox system. Okay, Cre-lox system is a system which is used to, suppose example, this is a DNA. In this particular DNA, this DNA is made up of many group of genes. Now I want to isolate a specific gene. I want to take out a specific gene, right? Which means first I have to identify the location of that gene. So to identify the location of the gene, to delete the gene, to replace the gene with the old one, or to invert the gene, means changing the position of this gene. So in simple words, to detect the gene, at the same time to delete the gene or replace the gene with another gene, or to invert the gene in a DNA, we use this particular Cre-lox system. Now the question they are asking is, which of the following options is correct about this particular Cre-lox system? So generally friends, in case of a Cre-lox system, see here what we generally require. We require two things. One is Cre-recombinase enzyme. If this Cre-recombinase enzyme want to show a certain function, it will go and associate it with a specific site on the DNA, named as what site? LOX P site. So if the Cre-lox system wants to work, definitely what do we require? We require a recombinase enzyme and we require LOX system, right? Wherever these two are present, then deletion, inversion of the gene will happen very easily. And where this particular LOX P sites are present, on the DNA it is present on the 5' sites also, as well as it is present on the 3' sites also. If the, suppose example, this is the Cre-lox site, now automatically recombinase enzyme will come and associate. By that we can delete the gene or we can replace the gene. So mice containing LOX P sites on both 5' and 3' ends of the gene were mated with mice that are transgenic for Cre-recombinase. So you remember, if the Cre-lox system wants to work, recombinase is required and their binding site is also required. So wherever both are present, then the recombination or inversion or deletion of the gene will happen. It is the simple one. Here they are saying LOX P recombination sites were introduced in a tandem at the 5' end of the gene. What is called tandem? Very good. Which is in a repeated. It should not be in that manner. It should be in a flanking manner. Flanking means what? They are present on both ends in a hanging manner, on 5' also as well as the 3' also. So LOX P recombination sequence were introduced only at the 3' end. It is wrong, right? It should be present at both 5' as well as 3'. So the most appropriate answer is 4. See, there is no explanation here. I want you to remember compulsory. Okay, it is all fact based. Which one of the following phytopathogenic bacteria is predominantly transmitted through the insect? I want every student has to remember Xylella fastidiosa. Please remember, there is no exception over there. Xylella fastidiosa is a phytopathogenic bacterium causing diseases like a Pierce disease in grapes. It's notably transmitted by xylem feeding insects like sharpshooters, right? So just remember name of the insect as well as on what fruits it is acting, what is the name of the disease. That only I want you to remember. Whereas Xanthomonas oryza, it generally transmitted through the water. Ralstonia solanaceum, it also transmitted through the soil as well as water, and very few times insect, very few times. Pseudomonas, it is also transferred to the rain, aerosols or infected seeds. But the question is specifically asking about specifically through insects, which is Xylella fastidiosa. You have to remember that. This is also a straightforward question only. See, in simple words, one organ is going to decide the fate of the other organ, or one area is necessary for the development of the other area. In other words, one area is going to influence the fate of the other area. That is called as ectopic expression. So in very simple words, friends, if the wing wants to be developed, what is generally happening? The normal structures such as developing eye, antenna, leg disc, all are converting into what? Wings, right? So this is influenced by what? This is influenced by the vestigial structures. So vestigial expression is necessary or essential for wing development. Okay, you remember that very correctly. Vestigial expression is always required for the wing development. The same question was asked in the 4 marker also. You just remember that we will answer that. This is again 11th standard question, simple question. You remember, anyone remember in the yesterday class also this question came in the set exam, the same question. Options also, they just given a word cistron, that's all, in the set paper. Which one, what is the answer? Which is what? In case of a normal bacterium, it is having peptidoglycan, whereas in Archaea it is having pseudopeptidoglycan, or we generally call it as slime layer. Right now, this is the one, right? Absence of nuclear membrane, presence of single circular chromosome, presence of multicistron. The question is differentiate, remaining all are the common features. The only difference is bacteria contain peptidoglycan, Archaea contains pseudopeptidoglycan or slimy layer. Straightforward simple question. You remember the same question we have studied, what we studied, you remember h square, which is height. If you recall, in the given question they have given 0.8% and in the options they have given 80%. Remember the same question. Remember, it is a straightforward question. Which one of the following relationship correctly represents the narrow sense heritability? You always remember it is a fixed formula, there is a no change in the formula. Additive variance by total variance, right? Suppose example, what is additive variance? It may be anything, it may be a height, it may be a color, it may be a shape. Variance of a single trait by variance in the total variance. Variance of a single trait by total variance. Simple question it is. You remember the formula directly, VA by VT. This is also very simple, straightforward, a 2 marker. So far, have you answered at least 4 questions correctly? Everyone? Jayanti, what is the answer? You all forgot. The activation of CDK2 in vertebrate is dependent on. How many times we read in the slide? Others, what is the answer, everyone, online people? That's why you are thinking that much of time. We already studied, right? CDKs will work in a two ways. One is CDK combines with cyclin, another phosphorylation and dephosphoryl. Correct or not? So, but cyclins are always having what? Lower lifespan. Cyclins will come and activate the CDK. After the cyclins are getting degraded. You remember CDK is having 2 binding sites. One stimulatory tyrosine and inhibitory tyrosine. Remember or not? Association with what? Cyclin A. Simple question. This also we read. We read a test. You remember cis-trans test. Very, very good. Which is called as complementation test. Wonderful. Very good. I have explained body color of the insect. Everyone please answer the question. I have explained it so many times. If the mutations are present within the same gene, they never be complement with each other. You all forgot, not reading at all. Okay, imagine this is A plus gene, right? This is also A plus gene. This is a B plus gene. This is also a B plus gene, right? Imagine from here one copy is coming. Okay, otherwise let's consider like this. A minus, A minus. So now from here we get one copy, one is mutated and one is normal. Correct na? Here also one is mutated, one is normal. Here complementation never happens, right? But when the complementation is possible, A minus, A plus, B plus and B minus. Now from here we get one mutated copy, here we get one normal copy. So what is the genotype? What is the phenotype? Which is wildly type? Can I say that A plus? Because A is the dominant. Same thing will happen here, B minus and B plus. Which is B plus, which is a normal one. Correct now? Which is the wild type. So what is the point? All the students look into this. If two mutations are allelic within the same gene, it's the same line only we read, they won't complement with each other, which resulting into no wild type. Correct or not? So now what is the answer? Huh? Allelic or non-allelic? If they are non-allelic, then complementation is possible. You are getting the point. In this case, complementation is possible. Here, in this case. Consider these are two minuses. Complementation is possible? No. Complementation is possible when they are non-allelic. See, simply remember, complementation is possible when they are non-allelic. Complementation never be possible when they are allelic. Now what is the question? Two temperature sensitive mutants of E. coli were mated with each other and none of the resulting X conjugates showed the wild phenotype. If they are non-allelic, definitely they show what? Wild type. If they are allelic, they won't show it. Right? Answer is allelic. In the extension of the Mendel principles, we have read this complementation test and I have explained it. This also we know it. It is also direct there, it's there in the slide. Understand everyone previous question? Everyone understand question number 20 online? What's the answer? These are there directly in the slide itself. For a given organism, which one of these life history attributes are associated with the broadest range of environmental tolerance? What is the answer? The question is very clear. Among these characters or attributes, which is helping the organism to tolerate to the environmental condition? Suppose example, if the organism is tolerating the environmental condition, what that organism generally called as according to the Darwin, or we call survival of the fittest. Now they are asking what is that, which is obviously survival only. Correct na? See, these are all there in the slides, so you check it correctly. This is what? Survival. This is also a question from the extension of Mendel principles. We read a concept, if everyone quickly recall, incomplete dominance. After that, we solved something called as calculating the number of genotypes or number of gametes. Third or fourth slide of extension of Mendel principle. Formula is 2 power n. I have given the formula, no? Tell me, what is the answer? Drosophila melanogaster has 4 pairs of chromosome. there is no recombination in males of this species, what is the number of possible combination of chromosomes in gametes? Correct na? You always remember friends, for the number of gametes, the formula is what? 2 power n, right? So they have 4 pair of chromosomes and no recombination also occur, right? Which is 2 power n, 2 power 4, which is 16. We read a concept named as multiple allelism in the extension of Mendel principles. Use the brain. Slow. That much fast, how man? Okay. See, this is the wild type. Wild type contain AT, GT, AT, CGA. Correct na? So this is one codon, this is second codon, this is third codon. Right? There is a mutation. So it is a wild type, which means definitely it can code for a phenotype, which is functionally active. There is a small mutation occurred. The T got replaced with A. What are the 3 codons present in the mRNA? UAA. Similarly, DNA also having the same kind, right, which is opposite. Among those, one of the codon is what? TAA. So because of mutation in the T, it has given rise to what? A. So what codon it is? Stop codon. Then what happened? Entire information is losing. Correct na? So which is loss of function. Clear everyone online? Okay. So this is the wild type TAT, code for tyrosine. The T is replaced with A, which is coding for stop codon, which leads to premature termination, which obviously leads to the loss of function. This is very important question, clinical question it is. The question is, level of which of the following parameters of blood are measured to check cardiac damage? So we have variety of enzymes. Which one we can eliminate easily? Urea. Correct na? Urea do not have any role in terms of cardiac damage. What else we can remove? Peroxidase. Is it? Then there are only 2 options. One is the creatine as well as the creatine kinase. Generally we use the creatine kinase. To assess cardiac muscle damage, clinicians commonly measure the total amount of the creatine kinase. You have to understand the point very clearly. Creatine kinase won't be released all the time. Creatine kinase will be released into the blood only when the heart muscle cells are damaged. When creatine kinase levels are increased in the blood, which indicates that myocardial infarction. Right? Suppose if I want to measure this urea, what does it indicate? See, here we use creatine kinase as a marker for the heart damage. Why we use this urea? To measure the kidney function. Correct na? So this is the kidney marker. Whereas peroxidase is not at all directly or indirectly not associated with the heart. Right? Whereas creatine kinase is the major enzyme which is which level will be increased upon muscle damages of the heart. It's a simple question, clinical question. 1 second. Which one of the following does not promote aging? They are asking does not. You know it everything clearly. Activation of telomerase. Correct. Now what is the function of a telomerase? Telomerase is the enzyme which increases length of linear DNA. Correct na? So if telomerase is present, DNA length is getting increased. Correct na? Which means lifespan will increases. If telomerase is absent, length of the DNA will decreases, then lifespan will decrease. So, but they are asking which one of the following does not promote the aging in humans, which is activation of telomerase. Simple. Others, everyone online? Triad means there are 3 amino acids are there. So, we have histidine, we also have the serine. Now, you have to understand the relationship between these two, by that you can understand. What is the side chain of serine? Very good. Histidine, imidazole ring. Anyone remember histidine whose pKa value is near to the neutral, which is nearly 6. So, it can act like an acid also as well as the base also. Now, based on that you tell. It's very simple. Look at this. First look into this. Histidine whose pKa value is what? 6, which is near to the neutral pH. So, it can act like an acid also, act like a base also, right? It pulling a proton away from the serine. As a result, what is happening? The serine, upon losing the proton, it is converted into what? Serine alkoxide. What is that? Serine, we can say alkoxide, which is the highly reactive nucleophile that attacks the peptide bond, right? It's very simple point here. If the serine, I mean to say whatever the electron that is, whatever the proton that is associated with the serine, if it wants to be pulled towards this particular histidine, the value of the pKa should be always lower. In more simple words, serine has to undergo what? Deprotonation. You are getting? If it is ready to undergo the mechanism of the deprotonation, histidine can easily pull the proton away from the serine. So, the serine side chain must have lower pKa. This makes it easier to lose the proton. Upon losing the proton, automatically what happens? The pKa value is also getting decreased, right? So, the pKa decreases because the hydrogen bonding with histidine stabilizes serine side chain in what state? Deprotonated state. So, in more simple word, histidine being a base, it is pulling the proton from the serine. It loses the proton since its pKa value is lower. More simple. After that, read the explanations and remember one more time. This is straightforward question, which you read in bachelor's and master's also. Deepesh sir also explained it the same. It's very simple, you remember. If you want to observe any transparent specimens, if you want to observe any transparent specimens without staining, if you want to measure any, observe any transparent specimens without staining, such as, the C. elegans, we use a special microscope, differential interference contrast microscope. Please remember that, okay? Because it is highly, suitable for the transparent specimens. We do not require the staining, and it is completely non-invasive method. Okay, remember that. Remember, they may also ask you the question, among all of the following microscopic method, which microscopic method can be used for the non-invasive methods? That is also differential interference contrast microscope. Remember, everyone online. See, this is actually there in the standard reference books from the concept of host pathogen interaction about the papilloma virus. Now, commonly remember papilloma virus, the first point, the viral structure protein, first you generally remember papilloma virus generally causes the cervical cancer, okay, cervical cancer of the epithelial cells. This is very clear, right? And option 1 says that the viral structure protein encoding genes occasionally integrate into the host genome leading to the cervical cancer. It is not at all occasional integration, okay. After the normal integration only it starts acting like a oncogene and it stimulates the tumor formation. So remember a common point, the viral structure protein encoding genes are not occasionally integrated. Then the viral protein E7, the viral protein E6, all the students remember these two are act like what? Oncogenes. Now let's quickly recall, here we discussed about in the cell biology we discussed about RB protein. What is the specialty of RB protein? It is going to block what? E2F. What happens if the E2F got blocked? Do you think cell division occur or not? Division never takes place, yes or no? But in case of a cancer, what happen? The cell division will be more than the normal. So E7 and E6, they never ever promote the RB. You are getting? E7 and E6 generally act like a oncogenes. Okay, these two statements are irrelevant statement. They infect the cervical epithelial cell and maintain themselves in a latent phase of extra chromosomal plasmid. It's a fact based we have to remember. Please check at least you will be able to answer 15 to 20 questions or not. This you know, everyone knows very well. Slowly, okay. Easily you can eliminate. So you have to arrange highest to lowest. Generally cholesterol present highest where? Plasma membrane, so we can eliminate these two. Definitely plasma membrane. After that, see, I have told you so many times, especially in the cell biology when we are discussing mitochondria, inner mitochondrial membrane is lack of cholesterol. It contains a special lipid. Anyone remember? Cardiolipin. Remember? So cholesterol is completely low. Where? In the inner membrane of mitochondria. This one also you can eliminate easily, right? So first it is in plasma membrane, and again most of the students think that since cholesterol synthesis is happening in the smooth endoplasmic reticulum, which means the cholesterol presence is more in the smooth endoplasmic reticulum. No. First it is higher in the plasma membrane, lysosome, smooth ER, followed by inner. So in the plasma membrane percentage is 30 to 40, right? We can say 15 to 17 in the lysosome, right? Whereas smooth, in case of smooth ER, low cholesterol, almost no cholesterol in the inner membrane of the mitochondria. Instead of that, it contains a ether linked lipid named as cardiolipin. The heteromeric G protein signaling component that is uniquely present only in the plant. We already discussed about the G protein. How does the G protein look like? G protein contains what? G protein alpha, beta as well as the gamma. Okay, this is alpha. Right. There are only 3 domains. Which domain associated with the plasma membrane? Alpha and gamma. Correct na? Now the question is here, among the G protein of animal, how it is different with the plant. Now remember, the G protein alpha in case of a plant having long N-terminal domain compared to the animal. So it is having extra large G alpha subunit in their N-terminal domain, which you have to remember. These are unusual G alpha proteins contain an N-terminal long domain, which is not found in the animals. Please remember that. This is also core botany question of intermediate level or degree first year you might read. Now what is clisteogamous and chasmogamous? The flowers which do not open. Correct na? These are the flowers which are open. It's very simple. The question is why might. It's not always true. Why might a plant species evolve clisteogamous in addition to the chasmogamous? I'll tell you in a very simple words, it is always a backup strategy. In more simple words, if the plant wants to produce chasmogamous, it requires certain environmental conditions. Suppose that suitable environmental conditions are absent, then that time they start producing clisteogamous flower. Okay, it is always a backup mechanism. Some plant species produce both clisteogamous and chasmogamous flower as a reproductive strategy, right? So we can say to increase the reproductive success in response to uncertainty in the pollination or uncertainty in the environment condition. Producing clisteogamous flower act as a backup strategy to ensure reproduction when pollinators are unreliable or environmental conditions are fluctuating. It simply, remember, it's a backup strategy for the process of pollination when the environmental conditions are not favorable. We already read it now. Simple. A equal to T, Z equal to C. Quickly. Now what is the answer? 1 na? Correct na? What is the percentage of adenine? 19. Thymine also 19. Then total how much it is? 38. Then 62 is left out. Cytosine and guanine. This is also 12th standard NCERT in the first lines. These are in the first slide of our PPT is also there. I mean to say in the third, fourth slides. In the starting I explained. Everyone, online people give the answer. According to the apparent Haldane hypothesis, which one of the following statements best described as Earth early atmosphere? We called prebiotic atmosphere and early atmosphere. What is the difference? Only difference is oxygen. When oxygen is present, that is only called as early, I mean to say new atmosphere, right? So it is reducing atmosphere with gases such as methane and ammonia. Correct na? Simple. Straightforward question. These are also there in the slides. Remember anyone? It's also there in the slides itself. Online, what is the answer? It's a straightforward question. It's there even in the slides also. Which of the following chemical modifications of uridine residues occurs after transcription of tRNA? We know very well in case of a prokaryotic organism, mRNA undergoes post-transcriptional process. Similarly, tRNA also undergoes post-transcriptional processes. Is it? Exactly at the T loop. Remember? So, when we discuss whenever the tRNA got synthesized, it undergoes a clover leaf folding and it is giving rise to stem and a loop-like structure. You remember or not? One is called as, remember, anti-codon loop, correct na? One is codon, remember or not? And another ones are stem. Especially at the T loop, this particular thymine undergoes variety of modifications. See here, there is a formation of ribothymidine and there is a formation of pseudo-uridine. Ribothymidine is formed by the methylation of uridine at the 5th position. Pseudo-uridine formed by the rotation and reattachment of uracil base to the ribose. Simply remember, tRNA undergoes two modifications during the post-transcriptional process. Correct. Ribothymidine and pseudo-uridine formation. Simple. It is there in the slides. Next. The question is, which one of the following is not a microbe associated molecular pattern of phytopathogen? What do you mean by that? Among these four, three are released by the microorganisms except one. See, these three are released by the microbes. This is the only substance which is released by the plant. Okay, now remember, you ought to remember, write down if possible. See here, microbes associated molecular patterns here are the conserved microbial molecules recognized by the plant innate immunity. So, FLG22, EIF26, PEP13, these three are released from the bacteria. Whereas cystamen is the only one not released by the bacteria, but it is released by the plant. It's simple. Great Indian bustard, which is commonly found in the desert grasslands. This is also a UPSC question, okay, which is commonly asked. You ought to remember, it is there in the book which we have to recall. This madam has, Vasiki madam has explained in the class. See, they are giving based on author's choice. In simple question, which one of the following organism lineages under the ICN or under the ICNP based on the author's choice, right? Which is what? Cyanobacteria. It is blue-green algae. It's a fact-based which you have to remember. There is nothing to explain over there. This is also there in the PPT itself, straightforward question. I believe everyone answer. Simple question, we have studied this. Remember, eukaryotic transcription, straightforward question. What's the answer? What is the first transcription factor? Transcription factor 2D, made up of TBP, TAF. Very, very good. Correct na? TBP associated with Tata box. TAF associated with INR, which is the initiation region. Then what is the next factor? Transcription factor 2B. What is the function of 2B? I have written a line also. What is that? Tell me correctly. It maintains the stable interaction with the TBP as well as the RNA polymerase 2. Right? It's a simple, straightforward question. It is there in the slides only. You're not reading. I don't know. You're not answering that I am here, or you don't know the answer really. It is very straightforward question only, no? Others online also, why you are not answering? It is the first slide that what we discussed. It's okay. All of the following are covered by lipid monolayers except everyone. Everyone. Monica, what is the answer for this question? 41. All of the following are covered by lipid monolayers except, means there is one substance or a molecule which is double layer or bilayer. Check it. Others. See, here chylomicrons as well as the endocytic vesicles. These two are kind of contradictory. If you look into the chylomicron is originally single layer. Inside of it, it contains phospholipids and it contains proteins. Understand all the students very correctly. When you observe the structure of the chylomicron, mostly it is a single layer, but inside of it, it contains lipids as well as protein, right? So chylomicron is generally a monolayer. You remember what is called endocytic vesicle? Vesicles are generally derived from membrane, correct na? Suppose example, let's consider this is a macrophage. So this is the plasma membrane of macrophage you consider, correct na? The pathogen got trapped, it entered inside. We generally call what? Endocytic vesicle. This endocytic vesicle derived from what? Plasma membrane only. Plasma membrane is what generally? Bilayer. Correct na? So endocytic vesicle is the bilayer. Chylomicron is a monolayer. Very low density lipoprotein is monolayer. Lipid droplets are also a monolayer. So whichever is derived from the plasma membranes, they are the bilayers. So lipid monolayer, and this will be the bilayer. Oh, this also we read. Four is correct, but that is not appropriate. Most appropriate. What is the answer? Which one of the following protein is responsible for the permeability of magnesium through the tight junction? We discussed it, right? Tight junctions are made up of cladins, occludins, tricellins and jam. Very good. Correct na? Among all, the most major protein is what? Cladin, which is responsible for preventing the movement of ions. Here in case magnesium. So which cladin is responsible for that? Okay, which is cladin 16. Ten you answered so far correctly. Remember, nothing will happen even though if you don't answer 5, you try to remember. First you remember. This is very good question. First of all, what is called metabolic rate? people will say no, tell me this statement is true or false. When we drink a cup of water in the empty stomach, people say metabolism will increase. Metabolism is rapid. What's a general question? Of course, metabolism will be improved, okay, when we drink a water, because it filters all the particles, all the waste particles in the intestine and improves the metabolism, right? That's simple. Everyone, what is the answer, online people? Okay, look into this. Everyone please remember, what is the major rule or a principle that explains the basal metabolic rate? Basal metabolic rate can be explained by a Kleber's law. Okay, please remember the name of the law also. Compulsory, I want you to remember. Now see here, weight specific basal metabolic rate decreases non-linearly with body size. See, you have to remember metabolism as well as body mass are related with each other, or what? Related with each other. So what generally people will say, if the body mass increases, there is a possibility that metabolism is also increases in one condition. In another condition, if the body mass decreases, metabolism is also decreases, or vice versa, But this is not a perfect assumption. What we have to understand, weight is not the parameter that decides the basal metabolism. You are getting the point. Here what they are saying, your weight or a body mass generally decide the rate of metabolism. If it is increases, metabolism is increasing. If it decreases, metabolism is decreasing. See, you might observe, right, the people who are having higher fat content, they are also, they shows a greater metabolic rate. The people who are extremely lean, their metabolism rate is completely different. So what you have to understand, weight directly or indirectly do not show great impact on the basal metabolism. You understand? So weight specific basal metabolic rate decreases non-linearly with body size. If it is linear, which indicates that both are related. So weight and basal metabolic rate are not influenced by each other. That's what they always follows, non-linear fashion instead of the linear fashion. So always remember, it is non-linear with the body size. Simple. Nothing will happen, you try to remember. See, the urea cycle generally occur in the two compartments, partially in the mitochondria, partially in the cytosol. Okay, not completely in the mitochondria, not completely in the cytosol. Now they have given some products, and these intermediates, among these, which of the intermediates are crossing the mitochondrial inner membrane? Only citrulline and ornithine will cross the inner membrane of mitochondria, whereas arginosuccinate, it is associated with outer membrane of the mitochondria as well as in the cytosol. So the urea cycle occur partly in the mitochondrial matrix, partly in the cytosol. So in case of inside the mitochondrial membrane, what we can observe, there is a formation of citrulline. Then citrulline transported out of the mitochondria to the cytosol, whereas ornithine is transported into the mitochondria from the cytosol. So in more simple words, we can say citrulline go out of the mitochondria, whereas ornithine moves into the mitochondria. So which are related here is citrulline as well as the ornithine. It's a simple question, urea cycle. I think Manas sir also explained Ramachandran plot. It is so easy. Simple question it is. At times sir has explained the same examples only. Which of the following is the correct arrangement of amino acid dipeptides in increasing order of the area of allowed region in the Ramachandran plot. See friends, in case of a Ramachandran plot, you have to remember we always arrange them based on their rigidity, based on their flexibility. Based on only these two parameters, we will keep the amino acid in the map, right? So they are asking increasing order of the area. So we can consider rigid and flexible. You know which is generally least flexible or more flexible? Glycine, correct na? Because it contains only hydrogen bonds, right? So definitely we can eliminate these two. And definitely which should be the first one? Proline. Proline is the rigid because it do not have hydrogen bonding. It forms a group named as imino group. Correct na? So proline, phenylalanine as well as the glycine. So remember, the area of allowed regions in the Ramachandran plot depends on how flexible or restricted an amino acid is. So most restricted is proline, then followed by phenylalanine because of the presence of bulky group. Bulky residues of phenylalanine will be present in the middle position, followed by the alanine, and the least most flexible is glycine. So the answer will be the third one. Which one of the following biofuels are classified as first generation biofuels? Okay, see, you have to understand the point very correctly. First biogeneration biofuels are those type of biofuels which is exclusively prepared from food crops. Algae cannot comes under direct food crop, right? So food crops such as corn, wheat as well as sugar beet. If you look into the bioethanol, it is exclusively made up of sugarcane or corn. That's the reason it is first biogeneration biofuel directly prepared from the food crops. It's also a straightforward question, which is bioethanol. This is also directly we have studied 4 or 5 times. I have explained the same line to all of you in the questions. Everyone, what is the answer? Ribosomes attaches to the endoplasmic reticulum by via. Why it is there in the slide only? Remember, this is endoplasmic reticulum. This is ribosome. How does this ribosomes attach to this? Anyone? Someone said. On the endoplasmic reticulum, there is something called SRP receptor, signal recognition particle receptor. Recalled or not? And we have one more protein called signal recognition protein. Remember, it is having 2 domains. One domain it is associated with ribosome, another domain it is associated with receptor. By that ribosome can come and attaches. Remember the slide. So the translated nascent chain which carries a signal, that signal only SRP signal. It is there in the slide itself, which allowing the interaction with SRP receptor on the ER. That's what I am saying. You come here and read. Whatever you read at home, it is different whenever you read here. It's okay, fine. This is also straightforward question. Everyone, online people give the answer. Deepa, why you are not answering this previous question? Which one of the following molecules is transported into enterocytes from intestinal lumen by glutes? No, it is GLUT5. Sorry. Which is what? Fructose. It is also there straightforward in the slide, in the matching column. Glucose transport by SGLT1, galactose also by same. In the previous year they asked galactose. SGLT1, fructose will be by GLUT5. Lactose though cannot be transported. Before transporting lactose has to be break down into glucose and galactose. Again they will be transported via SGLT1 only. You read this also. Online, mutation of homeotic genes often result in which one of the following developmental defects in Drosophila? What happens when the mutation occurred in the homeotic genes? That's the question. Because of the mutation in the homeotic gene, one segment can be transformed into the another segment. You remember, see here, anterior axis cannot be converted into the posterior and posterior axis cannot be converted into the anterior here. What they are saying, mutation of one specific gene. See, you have to understand the point. An example, anterior can be coded by what? Bicoid, and posterior can be coded by nanos and caudal. Example, if I insert bicoid here, there is a possibility that anterior structures will develop here. If I insert nanos here, there is a possibility that posterior structures will develop. They are not asking anything about the transplantation or experiment. They are asking what happens when there is a mutation in the homeotic gene. Because of the mutation, it never shows its function, or it can be converted from one segment to the another segment. It's a simple question. Same type of a question in the form of diagram they have given in the 4 marker, we'll see. Homeotic genes are Hox genes in the Drosophila determine the, see here, identity of the segments. When homeotic gene is mutated, the segment develops correctly in position but takes on the identity of a different segment. So look into this. There are 2 homeotic or Hox genes, UBX genes and antipedia gene. If these 2 genes got mutated, lex starts developing in the place of antenna. If UBX got mutated, halters transform into the wings. Position is same, position is not changing. There is, but one segment is taking the identity of the another segment. That is what we can call transformation or homeotic transformation. This question is wrong. Once again we'll read this. 1 second. Question mentioned wrong. Just a moment. Slowly. These are all 4 markers. Everyone, right or wrong, give the answers. Spindle assembly requires the formation of a numerous new microtubules. To meet this demand, the spindle contains a large number of regulatory factors. It is there in the pathfinder at the end. A few protein or protein complexes are mentioned below. The question is very simple. In order to form the spindle fiber, what are the protein or protein complexes are required for the spindle assembly? Tell me which is not required for the spindle assembly. What is the function of dynein and kinesins? So motor proteins, is there any role? So you can eliminate one option. So which one we can eliminate? Others, remember, we require remaining 3 proteins. Please remember now the proteins which are required for the microtubulin. Number 1 called gamma Turk, tubulin ring complex protein. The next one called as argmin. Argmin is the protein which recruits the tubulin ring complex. Please remember this very correctly. You remember, now tell me, how does the spindle fiber can be formed? By microtubules. You remember, we have alpha microtubules and beta micro. These 2 microtubules become polymerized by tubulin ring complex. This can be brought by Augmin. I believe you all people remember, we read some kinases, Polo, Aurora B kinases. Remember everyone? We read Aurora B. What is the function of Aurora B? Phosphorylation of nuclear lamina. Correct na? As a result, what is going to happen? Nuclear lamina is getting breakdown. Here we have Aurora A. It is helpful in the microtubule nucleation near the chromosome. So in simple words, we require these three: Turk, Augmin and TPX2. These three are required for the microtubule assembly. Dynein is a motor protein which is not involved. These are all factual questions only. This is from the regulation of the heart. What are baroreceptors? The receptors which are controlling heart movement with respect to the blood pressure. There is one more question which is related to it. First let us solve that question, when after that we will solve this question. Remember this question number 54. Remember this. We'll come back to that. This is a straightforward question. You read it. We'll come back to that question. So now we started part C. You remember, out of 75 at least you can solve 16 or not. That's the goal. Minimum. Okay, no problem. Look at this. The following statements are made regarding vesicle formation, transport of vesicle and fusion of the vesicle to the membrane. What is the question? Says that vesicle, how it is getting formed, how it is getting transport, how it is going to fuse. Expressing a mutant ART protein. First of all, what is ART protein? ART protein is generally responsible for fusing of the vesicle to the membrane. Fusing of the vesicle to the membrane. Now the question is, what happened? The mutant ART protein is constitutively bound to GTP, but it is defective for GTP hydrolysis. Then what generally happen? See friends, ART protein having two forms, GTP form as well as the GDP form. In the GDP form it is an inactive form, in the GTP form it is active form. If it is present in the GTP form, it is always active. If it is always active, it is always fused to the membrane only. Then whichever will comes and fuse with the membrane, that fusion can be interrupted. Once again, don't get confused. ART protein will go and associated with the membrane only in the presence of GTP. But due to the mutation in their GTP hydrolysis, it is always associated with what? To the membrane. As a result, whatever the vesicle will come and fuse with the membrane, that fusion will be completely disrupted or interrupted. So remember, COP1 coated vesicles are only formed from their normal location? No. Now remember, ART protein always associated with the COP1 coated vesicle. If ART1 is constitutively bound, it never allow the COP1 coated vesicles not to fuse with the membrane. So what you have to understand, B and D will be the correct. Look at this. A GTP locked protein, ART, exposes its membrane targeting amphipathic helix and remain constitutively membrane bound whenever it get mutated. So B is true. ART GTP recruits what? COP1. If the COP1 wants to comes and attaches, this should not be constitutive. This should be GDP form. But here it is in the GTP form. As a result, COP1 coat vesicles cannot go and fuse with the membrane. Its membrane fusion will get disrupted. Right? Because this vesicle fusion can be blocked by the constitutive expression of the ART protein. Simple. So B is correct as well as the D. No confusion. It is a straightforward question which is there in the slides only, which you all know very well. Everyone. Oh, this is not O, it's D. Everyone. Straightforward question only it is. You all read it, no? What happened here? In the C2 cycle, what do we get? We get one molecule of 3-PGA and one molecule of 2-PGA we get. Yes or no? Where this 2-PGA got produced? In the chloroplast. From the chloroplast, where they enter? Peroxisome. Phosphoglycolate first converted into what? Glycolate. Correct, na? Then glycolate entered into the peroxisome. Correct or not? Now look at the option A. 2-phosphoglycolate formed in the chloroplast is converted to glycolate. So what we removed, how the phosphoglycolate converted to glycolate? By removal of phosphate group. What is the enzyme that is involved? Phosphatase. Statement is definitely true. It is all direct statements which are there in the slides only. So A is definitely true. Then what we can eliminate? Option 2 and option 3. Glycolate exists to chloroplast by, is it true? True? Very good. Glycolate, it's very simple. What is called diffusion? Direct movement of, from higher concentration to the lower concentration. Do you think if they want to enter into the peroxisome, they will show the mechanism of diffusion? Glycolate is what? Negatively charged. How does it can show the mechanism of diffusion? It definitely they require carrier or the transporter. We call glycolate transporter. Correct, na? So the statement is wrong statement. C. In the peroxisome, glycolate reacts with oxygen and give rise to what? Glyoxylate and releasing of H2O. C is right. Correct, na? Only serine, glyoxylate aminotransferase is involved in the conversion of glyoxylate into the serine in peroxisomal reaction. No, it is completely wrong. Right? So what is the answer? Fourth. Right. It's a straightforward question only. Why no one is answering? Easily we can guess this question. Easy it is. Now what's the answer? The following statements are related to the airways resistance of lungs in the humans. What is called airway resistance means? We are restricting this particular bronchi or the alveoli. Okay, now look into this. The resistance to airflow increases with increasing lung volume. My lung volume got increased. Then what happened? That extra volume we have to send it out. Correct, na? If we want to send it out, we have to do the process of what? Exhalation. Correct, na? So when the exhalation is happening, the resistance to airflow is getting what? Decreased. With increases lung volume. Option A is definitely a wrong option. See here. Airway resistance decreases as lung volume increases because most of the air is getting pulled out or releasing it out. Then what is the next option? The major site of airway resistance is in the first 8 generations of the airways. You can simply remember which are alveoli and terminal bronchi. Terminal bronchi and alveoli are the regions where the major air resistance will happen. The smallest airways contribute very little to the overall total resistance of the bronchial tree, that is also right. Breathing a mixture of oxygen helium instead of oxygen nitrogen result in the decreasing airway of resistance. Why? Nitrogen is soluble or not? See, when it comes to the concentration wise, nitrogen is the highest compared to the oxygen. Percentage of nitrogen is nearly 78%. We discussed Henry's law, right? What you have to understand, but nitrogen never show any effect in the lungs. Why? Because the solubility is extremely lower. Solubility is what? Completely lower. So you look into this. Breathing of an oxygen helium mixture lowers gas density, reducing the turbulent flow, and that decreases airway resistance compared with oxygen and nitrogen. That statement is also true statement. Now I will tell you in a simple words, you all people remember. Airway resistance always decreases. Remember, when there is a increase in the lung volume. First point you have to remember. And maximum air resistance will happen at the terminal bronchi as well as at the alveoli. Second point, okay. And third point, you remember, oxygen as well as the helium, they are going to decrease airways resistance. Remember these three general points. So this is generalized one, it is updated question. It's an informative question. What is the answer? Which one? See, as per the 2020 FAO Global Forest Resources Assessment, the climatic domains are ranked in descending order based on their proportionate distribution of global forest area. Number 1 is tropical, boreal, temperate as well as the subtropical. Whenever we discussed biomes, we have studied this. Okay. Remember, it is a factual question and simple question. At least one, if you are from microbiology or other, you could remember this, or pharmacology. It's called chemotherapy. It's lymphoma. At least one you can answer. At least you know what is Vincristine and vinblastin function? Anti-cancer. Is there any option that is suitable? Where is that? B is the one. There also you can eliminate some options. What you can eliminate? What one? 2 we can eliminate and 4 also we can eliminate. Very good. Now try something. So 1 is clear, B is 1. Think about shellac. See, shellac is an outer coat. We know, right, we are having capsules. The capsule outer coat can be covered by a shellac. It is coating for pills and food glazing. Then which is suitable? No option. this is correct only, na? Cognitive enhancer, Vincristine, chemotherapy, skin creams and moisturizer. Shellac is, where is this lanolin? Option A, options wrong. See, Vincristine though it is very clear, and shellac is also, it's very clear. I think options were wrong. ABC transporter, I think options are wrong. 1 second. ABC transporter, mate transporter. No. Options were wrong, or typing mistake, typo mistake happened. Here right only. I think options were given wrong. No. Lanolin and lanolin is a moisturizer. Options were given wrong. Sorry, I have typed wrong. This is also there in the Pathfinder. These options they have given here. Options were given wrong. It's okay. Now I want you to write down these 4 points. Write down quickly. Online people also write down these 4. SID1, SID2, PAD3 as well as the PEN3. Please remember friends, PEN3 is a type of ABC transporter. We know, right? ABC transporter is exclusively responsible for the transport of large molecules such as secondary metabolites. ABC transporters are never ever involved in entry of pathogen. It is generally involved in preventing the pathogen entry. So remember a point, PEN3 is a type of ABC transporter which is involved in preventing the pathogen entry. Now PAD3, it is the gene which is generally coding for cytochrome P450 monooxygenase, which is going to help the production of phytolexins, which are antimicrobial compounds released by the plant. Again is the pathogenic attack. No confusion, all of you write down. PAD3 is a cytochrome P450 monooxygenase code for this enzyme, which is responsible for production of antimicrobial compound from the plant named as phytolexin. SID2, it is isocarcimate synthase enzyme, which is needed to produce salicylic acid, which is a plant defense hormone. SID2 is the name of the gene which is responsible for producing salicylic acid, which is a plant defense hormone. SID1, which is also called as the mate transporter, which is we can say defense transporter it is. So remember PEN3, PAD3, SID1 and SID2. Please remember, there is a possibility again we can get the question. This is also a straightforward question. In the first time we discussed this, not during the revision. Oh. Anyone remember? Sandhya, can you answer? Sonic hedgehog signaling, anyone remember? It's okay, no problem. See, I believe at least any one fellow remembered. Sonic, see, this is the eyelid. This eyelid can split into two eyes or two bilateral eyes. The splitting of the eyelid into two bilateral eyelids can be stimulated by sonic hedgehog signaling. What is the question they are saying? Loss of function of sonic hedgehog. Then failure to split the eye field to form bilateral eyes. Then what happened? Which leads to the formation of a single eye only. Okay, I told you in the starting which is called cyclopia. Right. Then elevated level of the SSH in precaudal plate, which leads to the disruption of anterior neural tube specification. Loss of function of OTX, which leads to no eyes. Loss of function of MIT-F, defective pigmented retina. Now you write down, at least you try to remember these four also there in the slides, which you have to read. That's why we have to make small charts like things whenever we are reading, then only we will be able to remember. I'll come back to these questions. First to solve at least one or two you know, then you'll get excitement. A population of Indian wolf shows logistic growth curve. So you remember the formula? What is the formula? dn by dt Rn into k minus n by k. Now look at this. Everyone, online people answer. A population of Indian wolf shows logistic growth. Let the carrying capacity k be 200. So what they have given? They have given R. R is how much? 0.05. What else they have given? K. They have given 200. What they are asking? What is the maximum possible growth rate for the population in terms of individuals per time unit? Slowly understand, analyze properly and answer the given question. Before applying the formula, friends, first you have to identify the population size. You have to identify the population size at point K. That is very important. How can we calculate that n? When the population size is maximum, whenever it reaches half of its maximum carrying capacity. Suppose if it crosses its maximum carrying capacity, do you think population growth will happen? Above the carrying capacity, there is no growth of the population. Population growth rate becomes maximum when it reaches half of its carrying capacity. So what will be the carrying capacity? 200 by 2, which is 100. So n is how much? 100. Now you try. Now you apply the formula simply. Everyone calculate. Very good. Everyone. Very good, Monica. Correct. Substitute in the formula and tell me. Quickly. So what is the R value? 0.05. N is how much? 100 into 1 minus 100 by K is how much? 200. How much? 5 into 0.05. How much it is? 2.5. Correct, na? No, 5 into 0.5. How much it will come? 2.5. Right? Correct. So answer is what? 2.5. Answer the question. You all read it. We have solved even in the questions only. Everyone. We have studied in the lesson only, na 4, third. Third. Plasmalogens are what? Ether linked, right? The special ether linked is the plasmalogen. Correct, na? What is cerebroside? Galactose linked to the ceramide. There also we can come to the conclusion, right? Anyway, what are non-polar lipids here? Right, which are triacyl glycerol. Archaeal membrane lipids are, anyway, sphingomyelin means phosphocholine head group. Archaeal membrane lipids are what? Very long chain fatty acid. Very good. Answer is 3. Hey, what happened to the question? Okay. So out of 75, you can answer 16 to 20 only correctly. Okay. We no need to disappoint. What's the answer? 2. How come 2, ma? If you, this is the so simple question, no. So last will be what? Ligase. Correct, na? Is it about bacteria, na? Okay, not a problem. But we can see the options, no? Before polymerase 1, what will, polymerase 3, which will come? Polymerase 1 will come. Definitely polymerase 3. But we know polymerase wants to work, definitely it require primase. And anyway, they have given SSB protein, then helicase, gyrase. Michaelis-Menten equation. Remember what is Michaelis-Menten equation? V naught equal to, tell me what is the equation? What is the Michaelis-Menten equation? That time I have solved 2.5 hours. Everyone solved, no, that day.
V naught is equal to V max into S, right?
Km into S. That's all I am asking. Do it. A glucose biosensor X. That's all you have to apply the Michaelis-Menten equation. Everyone do it. You have to write down what was given. What was given? Km is given. 5.2 mm. What else given? Imax was given. How much it is? 250 microliter. And what else has given? Substrate also has given. How much it is? 3.4 mm. That's all. Calculate it. What is there in that? What is the current response of the biosensor? So I equal to I max, correct na? S by Km plus S, that's all. Calculate. Calculate, take out the calculators and do it. How can you do verbally? Okay, do it, calculator. How much? Very, very good. 98.83. Wonderful. That's all. Everyone got online people answer? 98.83. Very good. Now let's go back to the questions which are difficult. Okay. First, whatever the questions we know, we will answer. Then whichever you don't know, then we will go there. See, out of 75, how difficult the question paper it may be, how difficult it may be, 13 to 14 matching questions will come. You have to identify them very correctly. It's a 4 o'clock. Given below are the statements about the action of glucagon in the human body. Don't make it wrong at all. Everyone online? Monica, what is the answer? This much time, so simple question. Are the action of glucagon? Opposite functions of the insulin is glucagon. Why you are thinking this much of time? Which hormone is responsible for gluconeogenesis? Glucagon. Correct na? Glucagon majorly act on the liver or muscle? Major. Muscle? Liver. Epinephrine act on the muscle. Right. They are asking true statements. Who will do the process of glycogenolysis? Insulin or glucagon? Glucagon. Now, what is the first statement? True or false? In muscle. Is it true or false? It's true only, na. See, glucagon causes glycogenolysis in liver. So, it do not cause glycogenolysis in, so that statement is true only, na. Keep brains here. It does not cause glycogenolysis in muscle. It causes glycogenolysis in liver. What are ketone bodies example? There are 3 ketone bodies: acetone, acetoacetone, acetyl-CoA. These 3 are said to be what? Ketone bodies. It decreases the ketone bodies. Ketone bodies are generally formed by the mechanism of lipolysis. Now, glucagon increases the lipolysis or decreases? Increases. Correct? Now, it is involved in the breakdown of lipids. But what they are saying? It decreases ketone bodies. So, false. It does not cause lipolysis in the liver. It causes. So, this is also wrong. It inhibits conversion of enol pyruvate to the pyruvate in the liver. True or false? True. Why it is true? Glucagon generally activates what? pKa. Correct na? Protein kinase A. If this is present, what generally happen? What is the level of the glycolysis? Is it low or high? Level of, see, pKa is present, level of the glycolysis is high or low? It is definitely lower. Glucagon is show its function by the pKa. If the pKa is present, which means what is present? Glucagon is present. If the glucagon is present, the level of the glycolysis is what? Definitely low. So, it inhibits conversion of phosphoenol pyruvate to the pyruvate. Correct na? If there is a production of the pyruvate, insulin growth will happen again. Correct na? I mean to say glycolysis will continue. So, this statement is true. It inhibits the conversion of phosphoenol pyruvate to the pyruvate in the liver. So, what is the answer? A and D. Why you forgot this small thing? It does not cause glycogenesis in muscle. It is true. Correct na? Because glucagon receptors are absent in the skeletal muscle, which is mediated by epinephrine. It decreases ketone bodies formation, which is false. Glucagon increases lipolysis, which leads to the formation of acetyl-CoA is one of the ketone body. Don't confuse. Okay, it's easy. Okay, this is also you read multiple times. Interaction among maternal effect gene and gap gene products are crucial for establishing embryonic domains in the Drosophila embryo. Which one of the following options does not? They are asking does not. Take your time. They are asking does not. This is called giant. This is bicoid. This is caudal. This is hunchback. This is cripple. Very good. Simple. You know it. You can also remember like this, bicoid, and what is the other gene? In case of anterior, hunchback. Posterior, nanos, caudal. Correct na? Can I say that bicoid activate hunchback? Yes or no, ma? Both are responsible for anterior axis. Nanos also activate caudal. Both are responsible for posterior. This information you know. Based on that you do it. They are asking does not. Online people, what is the answer? Everyone. The question is which does not show the correct interactions. Bicoid is what? Anterior. This one caudal, posterior. Correct na? This interaction is correct interaction only. In the middle we are having joint. So can I say bicoid is only expressed at the anterior, caudal is only expressed at the posterior. These two are going to suppress the joint. Correct or not? Because joint majorly expressed in the middle. So bicoid and caudal definitely suppress or repress the level of joint. So this is a true one. In that way you identify. Again, this is what again? Bicoid followed by hunchback, then followed by knirpel, right? Sorry, knirps. Knirps is what protein? Central. Whereas bicoid is anterior. Hunchback is also anterior. Bicoid definitely activate the hunchback. Correct na? Whereas bicoid also activate the knirps, but it is suppressed by the hunchback. Which means hunchback and crimps are opposite to each other. That is also true. Correct na? Only bicoid and hunchback are working together. Even though bicoid is activating the knirps, that is blocked by the hunchback. So this combination is also correct. Third one. We can understand na, hunchback is definitely going to repress the knirps or the knirpel. Knirpel is going to repress the joint, or opposite is also happening. So this combination is also a right combination. Now here you can understand the picture. Now you can observe the third picture and fourth picture very clearly. Hunchback can suppress the knirpel, at the same time it can also activate the knirpel. Knirpel can inactive or repress the joint, at the same time joint is going to repress the knirpel. Correct na? Now look at the fourth one. It is saying both hunchback and knirpel are going to repress each other, which is wrong. Here what we can observe, hunchback can repress the knirpel also, at the same time it activates also, but here it is showing both are antagonists. If this is right, here what we understand, knirpel can inhibit this, and at the here what we understand, both are showing antagonistic, but here it is showing one will be inactivating, one will be activating, which is opposite of this. So this will be the not correct combination. Don't confuse. Once again read the explanation, you will get it easily. Everyone has to answer the question correctly. You all read it also many times. Let me know if anyone having any doubt in the middle. We studied so many times. RAN is in the GTP form where? Very good. Where? In the nucleus. RAN GTP form is in nucleus. RAN GDP form in the cytoplasm. At least you know these 2 points, you remember this, based on that also you can answer. Option A is correct or not? The import receptor binds to the nuclear localization signal. Definitely true option. So what I can eliminate? Right? Next. Now try to eliminate others. The nuclear import receptor cargo complex is dissociated by RAN GDP in the true or false? False, because in the nucleus it is always in the RAN, and here nuclear import receptor cargo binds to RAN GTP in the, this is also wrong. In the cytosol it is in which form? RAN GDP form. The import receptor contains several low affinity binding sites for the nucleopode. So answer is what? A and B. Okay, right. Okay, clear everyone so far? Remaining we will discuss in the tomorrow class. Okay. So now I will share the PPT. Now whatever the questions so far completed, you have to do it again on your own. Okay. Now these 5 days you do only one thing, you try to remember the concepts. Most of the people forgot entire concepts. Now remember, for every unit take only 4 concepts, only 4. An example, if you are revising biochemistry, take only 4 concepts: carbohydrate metabolism, amino acids, enzymes and its kinetics, either nucleic acid or the proteins or the lipids. Only take 4 concepts and only revise them. Suppose if you are reading unit 3 molecular biology, definitely take post transcriptional processes, transcription, replication and operon. Only revise 4. Okay, only revise and try to remember. What I observed is you forgot almost all the information. Try to remember. There is no need to demotivate. We cannot answer every time all, and most of the questions you couldn't answer because you didn't remember anything. Okay, you have to remember while you are solving. Remembering is most important rather than reading. Okay, nothing will happen. You revise every single question. In the tomorrow class I will complete this. Okay, clear everyone? No, Deepa, it is not fine to answer 10 questions. No. In case if you are a OBC candidate, out of 75 questions, example I am saying, if you are a OBC candidate, consider I am using the word maximum. I always use the word maximum. 85 to 90 marks you have to get, 90 marks. In the 2025 June for OBC it is only 80 marks is the cutoff for the lecturership category. If it's a JRF category, it is 85. Suppose if you are lecturership, so how much you have to get? 80. Correct na? In that case, I am always using the word least. So cutoff was 39.46 or something for the OBC. Then that time how much we have to answer? Least only. In the part C, let's consider least to least 14 out of 75. Let's consider here at least can't we answer 3 questions in the part A? 6. How much it is? 56 plus 6. 62. Okay. Out of 50 questions in the part B, can't we answer even 12 questions? Easily you can answer. So how many marks are coming? 86. Correct na? So you should always remember this category and you have to answer. Okay. Clear everyone? But always we have to be on a safer side. Nothing will happen, we can answer the questions easily with a calm mind. All right, I'll see you in the next class. Okay, thank you all. Thank you. Suppose if you are from the SC category, what is the cutoff?